I'm going to talk about specificity, so I thought I'd give you a very specific title, and I hope that by the end these words are all clear. Okay, so I think we can all agree that specificity is an essential part of any biological system. And when we think about specificity, I think traditionally from a structural biology point of view, we often think about structure and binding that comes from structure as a way of understanding where specificity can arise. So this can come from secondary or tertiary structure for nucleic acids or proteins. And for biochemists, you can define a binding specificity saying that, let's suppose I have some molecular species I that has an intended binding partner I prime. The concentration of that binary complex relative to everything else it could possibly interact with would give me a specificity value. Of course, in the systems that we are interested in here, that's not always the case. And I want to emphasize that there isn't necessarily a clear dichotomy. Karen talked about systems where there is structure that leads to condensation, and Ben talked about a system where you can have disordered interactions that still lead to binary complexes under some conditions, right? But in many cases, we're thinking about systems where it's not so clear that you can just go from structure to binding to a binding specificity. And in particular, what I want to talk about is thinking about slightly different contexts for specificity, which I think many people are starting to use in a loose sense, which is about partitioning or composition, right? What goes where, what's in, what's out, and how do we actually define that and think about this property, because it really depends on everything that's in a mixture in order to get that rigorously defined. And so there have been a couple beautiful experiments of late that I think really motivate this question. I'll highlight two. One comes from Ben Savari's group, showing that if you make mutations to disordered regions, it's possible to redirect disordered proteins or disordered regions of particular proteins into different condensates in vitro. It's also possible to design completely de novo IDRs that, when expressed in live cells, partition into specific condensates. So this example, recent example coming from Rick Young's lab, shows that you can design a variety of completely new proteins that are partitioning into these existing condensates as indicated by this fluorescently labeled protein here. Right? I think it's fair to say that we don't have a complete understanding of how and why all this works. And here there's a large language model that's hard to interpret that is used for this classification problem. But nonetheless, I think it's incredibly exciting that it is possible to target using disordered regions. And so this motivates what I want to talk about, which is thinking about compositional specificity and how we can go about calculating something like this. So if we think about a concentration of a macromolecular species I in some phase alpha, then I can define an analogous specificity here. That's the concentration of that species in phase alpha compared to everything else it could go into. And if I'm at equilibrium, and chemical potentials are the same everywhere, then this is related to this expression here that involves the excess chemical potential. So it's basically the part of the chemical potential that has to do with interactions between molecules. Okay? So to show graphically the way I'm thinking about this problem, imagine that we have a bunch of different environments. Environments are defined by compositions. Let my graphics not lead you astray. Not necessarily droplets, right? They're just some sort of composition that has various macromolecules. And I can try to put different species into these environments. And doing so is exactly what this chemical potential means. If I think about going down a column, oh no. Okay, thank you. I've given away all the exciting parts. In case you didn't catch all that, let me go through a bit more slowly. So if I go down a column, I'm thinking about what is in a particular composition and why it wants to be there thermodynamically. If I go across a row, it's a bit more interesting. It's asking where does a particular molecule want to go? Right, so the question here is, can we predict mu I of this concentration, which is a function, from sequences for a sequence space? And I think more interestingly, can we describe the complexity of this function? Right, so by complexity, what I mean is there are clearly going to be correlations here. Right? To what extent can I actually design or choose different macromolecules that will partition into different places differentially, right? And that has to do with essentially how complex this function is. Okay, so on the left here, I have my sequence space. So every molecule is going to live in the sequence space. I'm going to call my molecular species I. And what I want to do is predict this entire function. And the function is the chemical potential of that species in any arbitrary environment. Okay? So it seems like a daunting problem, and indeed it is. But I think that one reason that we might have some ability to make progress is that we all have this intuition that there may be only a couple things about molecules that matter for this problem. So we can call this a chemical latent space. Right? So that is a compressed representation of a molecule that describes perhaps some features as yet undefined. And I'm going to use the letter Z to describe this vector for any particular macromolecule I. Okay. So all of this, this idea can be formalized in the context of information bottleneck theory developed by my colleague Bill Bialek and coworkers. And the key word here is relevant. Right? So we're not thinking about just any chemical latent space. It's not coming from a guess or my chemical intuition. We're trying to pinpoint exactly what is necessary to know about some molecule in order to predict this entire function. So that means that we're going to have an encoder, which means that the goal here is to maximize the compression of some chemical representation, and then also some decoder that is going to attempt to minimize the loss. So we want to get this function as accurately as possible. And in doing so, we can think about this functional complexity of what the decoder is doing. So in a very tongue-in-cheek way, we can say that the left side is chemistry, the right side is physics, but we can't fully separate the two because the relevant chemistry here is informed by what you have to do downstream. Right? So this really has to be thought of as an end-to-end problem, and I can show you that. Okay, so I'm just going to show you a few preliminary examples. This is a work in progress to give you a sense that this idea is useful. And so to get started, we're going to use some molecular dynamic simulations of a chemically specific heteropolymer model. Right? So these are single bead per residue coarse-grained models. I'm looking at disordered proteins here. This graphic comes from the first of these models developed by Jitane Mittal's group. Essentially, for every amino acid type, you have some interaction with all other amino acid types, plus some screened electrostatics and backbone connectivity. There have since been a number of revised models from a variety of groups. I'm actually going to show work from Jarell Joseph's group, has a similar model, parameterized differently, but the spirit is very much the same. And what you get by doing this is you put chains in a box, you run an MD simulation, and you can compute equilibrium properties. Okay. To be absolutely clear, we are, and what I'm going to show you, we're not simulating phase separation per se. Right? We're going to have a box that looks just like this. Okay. So this is work done by postdoc Zhuang and a first-year biophysics graduate student, Bethany, in collaboration with Gautam Reddy, also at Princeton. And what they've done is to start looking at how to apply this theory to IDP mixtures. So imagine that I have a collection of sequences that I want to know how they behave together. That's what I just said. Each one goes through the same encoder, right? It's the same representation in some latent space, represented by Z. And this has some variable dimension here. I get to control this myself, right? So the lower the dimension of this vector z, the more compressed the representation. For Jarell's model and the others of this class, there's no N-C terminus distinction, so there's an inversion symmetry here. Okay. Associated, to describe a mixture, associated with every sequence, I have to have a concentration. So this is literally just the concentration of each component in the system. But I want to emphasize that once we go to this latent space representation, the concentration vector is defined over the latent space, right? So it's how much of each point in Z space is contributing to the mixture. All right, and then I have my decoder, and I'm going to predict the pressure. So if you sense a bait and switch, there has been, right? This is not chemical potential, this is pressure. The reason is that it's much easier from simulations to get pressure. It's a purely mechanical quantity, but we can use the Gibbs-Duhem relation to get back to the chemical potentials that we care about. Right, so I'm going to compute this by running simulations, constant number of molecules, volume and temperature, MD simulations, very small, no phase separation, and use that data set to train this model. Okay? Should emphasize that any valid decoder has to obey certain physical symmetries. For example, if I swap the order of these arguments, I have to get the same pressure. There are several other symmetries that must be satisfied. And I also want to emphasize that we have to do this end-to-end training. So what that means is that it's necessary to train an encoder and a decoder at the same time in order to get a useful latent space. That's largely because we are considering sequence spaces that are so gigantic that we have extremely sparse data. And so what actually allows us to solve this problem is the fact that there's some reasonable representation between, reasonable connection between sequence and what's important about that sequence that serves to regularize this problem. Okay, and we do all of this within an active learning framework that I'm happy to tell you about the details of later. Okay, so as a couple examples, we can take a system that I think we understand pretty well. So we can take basically a hydrophobic polar model. So these are copolymers, length 20, super simple, but we already have 500,000 sequences. And we're interested in mixtures, right? So a binary mixture, there are roughly 1 trillion of those. We're going to train on just binary mixtures. We're going to use a loss function that will become clear when we think about free energies in just a moment. Then we're going to test on mixtures of completely unseen sequences, right? And what you get from something like this is generically a plot that looks like this. So this loss here, there's some simulation uncertainty. That's just coming from the statistical noise of the simulation. And if you use a transformer, which is a particular machine learning technique that's extremely powerful for dealing with models of this type, you get improvement as you increase the latent space dimension. Okay. To give you a sense, in case this doesn't speak to you, of what this actually means here, this sort of loss gives you a pretty good correlation coefficient between what's predicted by the model and the pressure seen in simulations. And again, all of this is on mixtures of unseen sequences, right? So this is far out of set testing. You can consider simpler models, right? Say, what if I forced myself to use something kind of like Flory-Huggins? And we're actually being as fair as possible here. We're learning what the correct excluded volume is for this, but we're forcing all the interactions to enter in a pairwise form. It doesn't work as well, it's not surprising. But more importantly, it doesn't show a dependence on the latent space dimension, right? Which is basically saying that by trying to fit everything into a Flory-Huggins style model, you can learn effective chi parameters. That's what we're doing here. But there's no use going to anything in this particular case beyond a rank 1 matrix because the model is not able to use that information. You don't have to stop at binary mixtures. You can try this on mixtures of unseen mixtures of 3 species. And this is the transformer model, works really well. Right? So you're able to train on some simple systems and use this to predict pressures. If you do this, you can ask, what do these latent spaces look like? So this is the transformer 2-dimensional latent space. Each color here is showing an increasing number of phenylalanine monomers in this polymer, right? So here are the 500,000 points. Okay, so as you move in this direction, you are increasing average hydrophobicity. What's happening in the other direction? Well, this has to do with blockiness. It shouldn't be terribly surprising, right? As you move from top to bottom here, you're going from something that is entirely uniform distribution, blockiness with the sticky on the end, sticky in the middle, sticky on the tail. Right? So that can help you to understand what's happening in the second dimension. You can do this for lots of different types of systems. So here's one with charged copolymers. Again, at the level of 2 dimensions, you get something that looks qualitatively like what we had before, but there's a bit more symmetry here because of the charge symmetry. Okay, question is this actually useful? And so if we think about predicting phase behavior, which is one but not the only goal of this, you can take an in-component sequence mixture, put it into its latent space representation, so that's just the concentration vector over Z, and then you can integrate to get a free energy. Right? So this is straightforward, assuming that we're always at equilibrium, and then convexify the free energy to get a phase diagram. Right? So this is, just as a subtle point, this is why we have the loss function that we do. You care about pressure divided by concentration squared for this step. Right? And we want to get this right. Okay, so phase diagram. If I have a binary system with just 2 components, phase diagram depends on concentration 1 and concentration 2. The orange dots here are the prediction of the model, right? So whenever you see an orange dot on this phase diagram, it says that this model is predicting a single phase region. And then these are now really big direct coexistence simulations of the same coarse grain model to check whether the prediction of this ML approach is working. You can immediately see that these are homogeneous single phase. Okay. Start changing the sequences. You put different sequences into the model, and you can predict different phase diagrams. So here we get tie lines, and then you can go and check. You put a simulation of the overall concentration of the open circle. It phase separates, you get an inhomogeneity, and the triangles here are the concentrations of the 2 macromolecules that come from this simulation. You can do more interesting cases. Overall, we get quite good agreement. And I want to emphasize that these are non-trivial sequences, right? So this is one of these head-tail sequences. And if you look in the dilute phase is not super homogeneous either. So this is, I think, related to what Antonia and Sam have talked about already in the workshop. Okay, so that all comes out of these models. And again, there's never phase separation seen. It just learns something about a particular derivative of the free energy from these small-scale simulations, and it's able to get quite far. The last thing I'll just point out here is that you can learn some things from this unbiased perspective about what actually matters with respect to intermolecular mixtures, intermolecular interactions. For both the systems I showed you, if you look at composition, monomer composition, non-linearly transformed to give you the best possible correlation coefficient, versus some projection of this latent space, you get extremely good correlations, right? Composition is essential. If you think about blockiness, there are a number of different metrics that have been proposed. For example, SCD, the sequence charge declaration metric, which was proposed to describe single chains. you get a correlation, right? But it's not. But what we're finding here is not exactly what's already been predicted, and I think that's one of the potential benefits of doing this in a very unbiased way. All right. So to just sum up very quickly, I told you about a way of thinking about specificity that involves a latent space that picks out what's relevant for intermolecular interactions. We can use this to predict phase diagrams. I think more powerfully, you can think about relationships between sequences as you vary this latent space dimension, you're looking at higher and higher fidelity, right? So some things you can get right with very simple models. Other things, other aspects of what happens with these cluster distributions, you need more information about the interactions, and that comes out very naturally here. So what we're doing, I think I talked about systems that I think for the most part we have a reasonable idea of what they do, so we can do this on more interesting systems. Within the same models, human IDRome is something that's underway, so stay tuned. It's possible to design sequence mixtures de novo within this latent space, and we have some preliminary work showing how to do this for some slightly simpler models. But I have to emphasize that all of this is learning what is available in this coarse-grained model, right? So this is really a starting point for interfacing this with presumably sparse experimental data, but I think this will give you a reasonable starting point and a way of thinking about the whole system as one. So I just want to emphasize Zhuang and Bethany, who did all the wonderful work that I showed you. Thanks for your attention.
Thank you. Questions?
That was very interesting. If I translate your Z of low dimension to what I was trying to talk about yesterday, I think my talk had a Z with 3 components. One was hydrophilic outside, hydrophobic inside, the opposite, and then 2 blobs of hydrophilic. Could you use that to, and we have no detailed sequence data on this. Could you use this Z that I propose to test various proteins to see to what extent that kind of coarse graining, or Z, I forgot what you called Z, but your Z is correct for various proteins. And part 2 of my comment is I didn't quite see where the solvent was in what you were doing.
Oh, sorry. These to hit that one first. These coarse-grained models are implicit solvent.
But there is solvent, so there are concentrations.
There's a pair potential between the amino acid beads in those simulations.
Right, but the amino acid beads don't use up all the volume of the system.
I mean, they can pack as Leonard-Jones beads would pack.
But is the sum of the volume fractions of all your protein beads 1, or is it 1 minus the amount of solvent?
It's more the latter. If
okay, that's
choose,
good.
Right? Because
okay,
it's not the lattice
good.
model.
Okay. Thank you.
But yeah, but I didn't answer your first question, but I do think that maybe we should talk about it later. Of course, you can have lots of different latent space proposals. What we're going for here are the optimal ones, in a sense, like the most compressed that gets you the best answer. But you can always do a mapping. Yeah, you can map between them, exactly. And I think that's one way of, like that last slide I showed is a way of trying to get some intuition there.
Hey, Will, great talk. Can you just comment on the going to more biologically relevant situations with longer polymers and more than 2 amino acids, your coverage of the sequence space, you mentioned how sparse it was. Yes. Even for 2, it's going to get worse, and presumably computationally it's going to be very heavy to deal with because it's just the sheer numbers. Yeah,
okay. So regarding how much data it takes to train these things, it's a chicken and egg problem because it's not so much about the size of the sequence space, it's about the size of the latent space that you need to start getting a small loss, right? And you don't know the latter before you try, but you can self-consistently find out. So we are finding out. But I think a large part of this, understanding the complexity of these functions, is trying to grapple with that question. And I can assure you that for the cases I did show you, where there are roughly 1 trillion pairwise things, and we considered 8,000 points, right? So it's very sparse, but of course the numbers matter. As for different sequence lengths, I think it's just a technical detail. It's a non-trivial technical detail, but we can do it.
So yeah, my question is kind of related to what you already said. So so when you have experimental data, right, the amount of data that you need to, but can you also do, I think maybe your last point in your outlook maybe points to that, use pre-training to reduce the amount of data that you would require?
Yes. So the last point here is basically this pre-training idea. The hope is, there are various ways one could try to do this. We have some ideas. I think a lot of this depends on the quality, quantity, and the exact form in which such data are available. Right? The key thing that I should point out is what you need to train a model like this isn't saying yes, no phase separation. You need to have some sort of thermodynamic quantity, right? Like a pressure or a chemical potential or something like that. And so if you have access to something like that, then you can more directly incorporate it into the training of the model. If you only have the sort of downstream effect of testing phase diagrams, then you would want to take a different strategy. But that's the goal.
Lovely talk. Well, many questions for after the talk, but for now, back to the title, information bottlenecking. I was just curious what your perspective was that on from what you're learning. Like, are we trying to think about bottlenecking from sequence space to thermodynamic space? Bottlenecking from what a particular model of a sequence is like? You've explored a certain class of models and what they can permit, and Kyogo, Kawaguchi, and others have pointed out these coarse-grained force fields can only permit limits. So I'm trying to think where the information because there's lots of bottlenecking happening here.
Right. So the information bottleneck. This one. Right. So this is, I think, the cleanest. Again, there's different ways of thinking about it. The cleanest way, I think, is a map from sequence space to chemical potential function over concentration vectors. Given some data source, right? But here we're using a model where I think that the benefit is that you can go back and test exactly what the model would say if you did the full whatever simulation you're interested in, right? Like a direct coexistence simulation is what I showed. But that is the precise way in which I'm thinking about bottlenecking. Yes.
A question on, in addition to predicting pressure, have you also thought about maybe predicting radio distribution function or some other statistical measures of like maybe that characterizes the microstructure or even the dynamic properties of these condensates?
Sure. I mean, it's all there. And one could absolutely take the same approach. So I can maybe give you two half answers. There are perhaps other things that could be better than pressure for this purpose. Pressure is just the easiest one computationally, but something like a structure factor that gives you access, so related to the RDF that you mentioned, right? But if you go to the 0Q limit, then you directly learn about chemical potentials. So, but they're noisier, right? So there's some trade-offs there. In terms of entirely different properties of, in these small-scale simulations, you still get interesting microstructure, right? Like the systems are small, they're not phase separated, but you still have lots of other information that we're completely throwing away so far. So I think one could look at other order parameters, and that could be super interesting.
I'm wondering what insight have you learned from this game? Have you sort of pinned the mechanism of how the IDP realized the specificity?
So in the bit that I showed you right here, we haven't quite addressed that question because this is really only one half of the story if you're going all the way to predicting phase diagrams. You also need to answer the question of in order to make many phases, what dimension, if that's even the right term, what dimension of this latent space do I need? So like how expressive of a polymer model do I need to be able to make lots of different phases where I control the compositions? We've separately worked on that, not using these chemically specific IDP models, but rather using more toy models, prem-regressed models. And I think that I'd be happy to tell you what we found in that regard. But the idea here really is to merge both of those things to be able to answer precisely that question.
Yeah, that's really nice. I was wondering, so a couple groups like Creston and Larson and Tanya have a paper doing sequence full, the full sequence variability predicting properties like CSAT or things on larger, more complex sequences. Have you run any of the sort of tinier, simpler sequences through those sort of models to see the comparisons and what they might be missing or what?
Sure. So the first thing I'd say is that we're really interested in mixtures here, which I think is a slightly different direction than predicting properties for individual proteins. And it really motivates this way of thinking about the problem, because if your map is between individual protein and what that individual protein does kind of on its own, or like a phase diagram for that one protein, then perhaps you learn different things. I mean, maybe you don't, but that's something that we want to address, right? So that motivates this slightly different way of thinking about it. This is very much a work in progress, and we need to make comparisons to lots of existing approaches. What I can tell you is that again, by working within the self-consistent world of CG IDP models, we can test everything that we're proposing. We can also test other proposals on precisely those models, right? So you can take mean field predictions and test them on these models, and overall, we've seen that that there's room for improvement. Yeah.
One last question. Stephanie, you want to
Have a really interesting work. I'm just curious about when you prepare these mixtures at the beginning, you have this NVT ensemble. So how does the temperature play here? Because definitely these sequences have different critical temperatures, right? And then when you have mixture, how you choose the temperature to mix them or have a considerable effect?
Right. Okay, so thanks for asking. Temperature is fixed, and everything here was done at one temperature, right? One could, of course, introduce something like that as another adjustable parameter or something that one can tune in the chemical potential function, of course. But given that what we've done so far, it's been fixed, I think the predictions would not be useful going beyond the same temperature. But yeah, I think it's something else that could also be incorporated.
Let's thank Will again.